Good morning. Good morning, everyone. Thank you for joining us at Avanti's session here at MANA 2021. My name is Tucker Hallmark, and I'm the Lipidomics Manufacturing Manager at Avanti Polar Lipids. And today I have the pleasure of welcoming Dr. Julijana Ivanisevic to share some of her exciting work that she's been doing on targeted metabolomics, specifically lipidomics, using one of Avanti's lipid mixtures, Ultimate Splash 1. But first, a little bit about Julijana. Julijana heads the Metabolomics and Lipidomics unit at the Faculty of Biology and Medicine at the University of Lausanne in Switzerland, where she also serves as senior lecturer. She joined the university in 2015 following her postdoc work with Gary Sustak at the Scripps Research Institute in La Jolla, California. And currently, Julijana's team focuses on characterizing sex differences in metabolism, both at the metabolite level as well as the metabolic pathway level. And this is in health and with the onset and progression of metabolic alterations with age. And to this end, her group applies high coverage, quantitative mass spec-based approaches to metabolic phenotyping of human populations in collaboration with clinicians and data scientists. And in addition to all of this, Julijana serves as the vice president of the Swiss Metabolomics Society. So today we'll hear about some exciting new work from her group that's been performed in the areas of metabolic phenotyping and quantitative lipidomics. And with that, I'll pass this over to Julijana. And if there are any questions at the end or during the presentation, please feel free to write those in the chat and we can address them after the talk. Thanks very much, and I'll hand over to Julijana.
So, hello everyone. Thank you very much, Tucker, for this nice introduction. And I would also like to thank Avanti Lipids team for their invitation, and of course the MANA organizing committee for hosting my presentation today. So I'm very happy to contribute to this third edition of MANA conference with our recent work towards high throughput metabolic phenotyping of human populations from single step extraction to omic scale quantitative lipid profiling. And here when I say omic scale, I do not mean the untargeted lipid analysis, but I actually mean targeted. Oops, I'm sorry. Here I wanted to characterize targeted in purpose as an omic scale targeted profiling in purpose to actually illustrate the power of targeted analysis and also to actually push the limits of targeted beyond the traditional targeted analysis, but not only the limits of targeted, I would also say the limits of the untargeted profiling. And here here the keywords would be really the specificity and the sensitivity, particularly the sensitivity of the targeted analysis as compared to really the untargeted assays. And indeed, we can say that today the high coverage targeted methods are becoming a common practice in many different laboratories worldwide. And specifically in lipidomics, they can actually bridge the gap between these traditional targeted methods with which we are used to focus on the quantification of one specific class of metabolites, and on the other extreme, the untargeted analysis with which we always try to actually detect as many metabolites as possible. And as you will see later on in the method that I will present, with the method that I will present later on, you will see that we can actually measure many hundred and up to actually even several thousand lipid species across many different lipid classes. Here, more than 25 different lipid classes and subclasses. And the great interest actually of this type of targeted methods, it's not just their coverage that actually allows us for an in-depth investigation of lipid metabolism at the molecular species level, but also the fact that we can benefit from the enhanced sensitivity and the dynamic range for lipid quantification. And I would say here that the quantitative data are indeed a prerequisite for multiple clinical applications. And if we consider this very vast structural and functional diversity of lipid species, we can actually claim that these acquired lipid signatures at the species level really represent a gold mine yet to be explored to improve, of course, our understanding of lipid metabolism and its association with the disease onset. And here, actually, the ultimate goal will be to use this vast lipid diversity as a resource to improve the risk assessment analysis, so the disease risk prediction models, and the health monitoring in general. And while really these consensus concentrations for many diverse lipid species have already been defined in standard reference materials such as the NIST plasma in several ring trials, so the interlaboratory ring studies. Importantly, the biological reference intervals for the majority of complex lipid species, and not only lipid species, the polar metabolites as well, that are actually composing the plasma metabolic signatures, have not yet been defined. And these biological reference intervals indeed remain to be established across different populations worldwide. And in the context really of this problematics, we are involved in several human population studies here in Switzerland. So these specifically concern here the Lausanne population and the complete health and heart cohorts that have been recently established at the University of Basel. So with this really prospective studies of human populations, we of course wish to contribute to the establishment of these biological reference intervals for a very wide panel of metabolites, but also far beyond this. So these populations have been clinically very well characterized, and most of them have also been, most of the participants have also been genotyped. And now we actually wish to complement the gene data with the metabolic phenotyping data. And we hope that we will be able actually to perform really for example, the lipidome-wide association analysis, including the causal relationship analysis that will help us actually to see, to identify the lipid species and the sets of lipid species that are really tightly associated with the age-related metabolic alterations that actually precede the disease onset. And on the top of this, as I previously specified, our ultimate goal is also to really use this lipid diversity as a really resource to improve the current existing models for disease risk prediction. And for this, in order to really be able to acquire these valuable complex lipid profiles at the lipid species level, we have spent almost 2 last years on the optimization of the lipid analysis pipeline. And this concerns, of course, this starts with sample preparation and concerns then, of course, data analysis and quality assessment. And to warrant really the application of this detailed lipid analysis in large-scale clinical population studies, we have developed a single-step sample preparation that is coupled to targeted helic tandem mass spectrometry approach for complex lipid extraction and quantitative profiling. And we have here opted really for the single step preparation or extraction because it can be easily automated. And we have, because we actually really wish to maximize the reproducibility of the sample preparation. So not only its high throughput, but specifically also the reproducibility. And on the other side, for lipid analysis, we have spent quite some time on expanding actually the coverage of existing LC-MSMS methods, and of course on the improvement of the quantitative capacity actually of these methods. So prior actually to deciding to go with a single step extraction, we have of course evaluated its performance and its efficiency. Again, is the commonly applied two-phase protocols. And in this case, we have specifically focused on two single step extractions. So one with, called BUMI, so the butanol methanol mixture, and and using 100% isopropanol. And the performance of these extractions was evaluated, was compared to the commonly used two-phase protocol with MTBE. These methods were evaluated using the subset of approximately 300 different complex lipid species and polar metabolites as well. I will here present really the results only on lipid species, but you can see all the other, you can easily find all the other results in our recent publications that is cited below. So here on the presented spider plots, you can see actually the relative signal abundance compared to the signal recovered with the biphasic extraction using MTBE. So MTBE, biphasic extraction protocol that is here used as a reference in blue. So as you can see, actually the relative signal abundance of complex lipids across many different classes in isopropanol and BUMI extracts was actually greater or equivalent to the signal recovered with the Matias method using MTBE. And on the top of this, importantly, the MTBE showed really limited performance for the extraction of lysophospholipids and particularly lysophosphatidylenosytols. inositols. And following this work really specifically on sample, actually on the extraction on lipid extraction, the optimization of lipid extraction, we have further continued really with the, we have further focused on the maximization of the lipidome coverage with the HELIC MSMS approach. And here you can find some of the parameters actually of the LC method that I won't enter into details, but we can discuss them later on after the presentation if you are interested. But what I would like to specifically highlight here is that this work was done on the last generation TSQ Altis instrument with dual channel UHPLC that was provided by the Thermo Fisher Scientific. And I'm highlighting this because we actually here really took advantage of an extremely high scanning speed of this instrument. So we were able to go down to the dwell time of 0.3 milliseconds while keeping really the correct peak definition. And this was fairly important for the development of this specific HELIC method because, of course, as you can imagine, in the beginning of the gradient, many different species of neutral lipids will necessarily coelute. So I would like to highlight here that actually the power of HELIC to separate lipid classes according to their head groups was recently recognized as an added value for the development of quantitative methods, specifically because the internal standards will actually coelute with the endogenous lipid species that are being measured. And in this way, we will actually be able to cover indeed all these lipid species with their corresponding internal standards to allow really for the appropriate correction of matrix effects in the same composition of the solvent as compared really to what we can see to the issues actually that we are encountering when doing this in reversed phase chromatography methods. Here, on the next slide, you can see, I wanted actually to illustrate, to give you the idea about the gradient. And you can see here, I used for this the mixture of internal standards, so that Avanti has released last year. So this is the mixture that we are using for the stabilized dilution. And here you can nicely see how the separation is obtained according to the lipid class. And I would like to highlight here that this Ultimate Splash 1 mixture contains actually the 69 internal standards. To this mixture, we have added 3 additional ceramide species and 3 additional species of free fatty acids, simply to actually complete the coverage of our method. What is fairly important is that, that this actually mixture covers multiple internal standards for each lipid class with varied fatty acid composition. And in this way, it actually allows us to really correct for differential ionization and fragmentation efficiency, depending on the fatty acid chain length and the degree of unsaturation. Finally, this same mixture of internal standards was also used to evaluate the linearity of the MS response, so the MS signal. We have done this by preparing the dilution series of Ultimate Splash, and then the dried solutions were actually reconstituted in the same volume of the matrix we were interested in, so in this case plasma, with the isopropanol solvent. And using this, in this way, we could actually look at the calibration curves. I mean, we have developed the calibration curves of these 75 internal standards. And based on the linearity of their response, we were able to actually validate the application, I mean, the applicability of the single point calibration for the lipid quantification. Finally, in order to really expand the coverage, I mean, to maximize actually the lipidome coverage, we have decided to perform first the initial qualitative screen, which in this case, as you can see, includes more than 2,000 different lipid species. This initial scan is performed on the pooled, like a pooled QC sample. This pooled sample in this case is plasma, because I'm presenting you the results from plasma, but you can of course prepare a pooled sample of whatever is your matrix of interest. And so with this initial qualitative screen in 6 subsequent methods, we can actually cover this, we can actually screen for these 2,000 different lipid species in the pooled sample. Then we actually filter this data. You can see these different criteria on the screen that are used for the filtering. And in this way, we actually decide about the species that are really put in the final methods for quantification. We wanted here to be very, I would say, very strict, because we really wanted to finally quantify or focus on the species that can indeed be robustly detected in the pooled samples. Here, I presented the results of plasma, so you can see that our final method for lipid quantification in blood plasma contains actually 548 transitions in positive mode. So these transitions cover the same number of lipid species in the positive mode, and then 284 transitions and the same number of lipid species in the negative mode. It's important to highlight here that the runtime is only 12 minutes, and combining both positive and negative mode. And this was possible, it was possible to achieve this actually using the dual channel UHPLC. In this way, we could actually multiplex the analysis on chromatographic columns, in this case, the 2 columns. And while actually the first analysis in the positive mode, as soon as it finishes and the column starts to recalibrate, we could already start the run in negative mode, and in this way actually save quite some time that needs to be dedicated to the column recalibration. On the bottom of this slide, I have provided several references that we have used to actually complement this list, I mean, this theoretical list of 2,000 species, which we have included in the initial qualitative screen. So this screen can, of course, be further adapted depending also on your interest. Here we have focused mainly on human biofluids, so plasma and urine, and also different types of blood cells. Finally, here I wanted to show you actually the coverage of our quantitative method, but the coverage based on the NIST reference, so plasma materials. And as you can see, with our method, we can really cover a very high diversity of lipid species across many different classes, including all different subclasses of phospholipids. And overall, we can measure more than 820 lipid species, combining the analysis in positive and negative ionization mode. So to further actually evaluate the robustness of our analytical methods as a proof of concept, as I have already specified, we use the plasma materials with different clinical and ethnic background. So these different actually NIST reference materials. Here you have all the information on these different materials, but in addition to the plasma reference material 1950, we have also used the pooled plasma from the diabetic patients, from the hypertriglyceridemic patients, and the young African Americans. And as you can see on the PCA plot, PCA score plot, you can see that with our method, we could nicely actually distinguish between these different plasma materials, because they indeed have quite a significant differences in their lipid composition. And here I will present you several circular plots on which we wanted actually to plot in more details these differences. So here you can see the differences of the material that was collected from the diabetic patients compared to NIST 1950 reference material. So you can see many different actually lipid species across many different classes that are being more or less highly abundant in this pooled sample coming from actually diabetic patients compared to the plasma to the reference material that was a priori collected from the presumably healthy patients. On this plot here, you can see the differences for the hypertriglyceridemic patients, and so you can indeed see a much higher concentration of tags that was easily detected in this material. And on the other side, you can also see actually that all of these different tag species are significantly significantly less abundant in the young African Americans, also because I guess these are really young participants compared to really the plasma reference material that was collected from the participants that are already above 40. And we know that lipids in general do enhance, lipid concentration enhances in plasma with age. Finally, as the proof of principle as well, we have analyzed the cohort of healthy aged individuals. This cohort was established here at the University of Basel, so it's a complete health cohort. I wanted to highlight that these are healthy because these patients were really clinically very well characterized, and they health, if you want, was evaluated based on several different anthropometric parameters, physical activity levels, and also biochemical parameters. Also, their cardiorespiratory fitness was evaluated really as a vital sign of their health in general. And as you can see on this circular plot, we can actually really claim that plasma lipidome is sex specific. This is definitively not the first time this is observed. This has been observed in many other studies previously, but I think it's not. I think unfortunately it's not well known and well recognized, and we are not paying attention enough to this, especially in the context of different clinical research studies. So you can see on this circular plot that actually many of these lipid species, specifically in the class of sphingomyelins and phosphatidylcholins, are actually significantly highly abundant in females, in aged participants compared to males. And this actually, I would say, positive association of lipid species with female sex has already been reported in several different studies. I have cited here two of them that you can look into for further details. But several of these, I mean, many of these species have actually been reported as being beneficial for overall health, because they were found to be negatively associated with the risk of development of specific cardiometabolic diseases, so such as obesity and different cardiovascular events, diabetes, etc. And finally, before I finish, I would just like to also put the accent on the need for the correction of the isotopic overlap. And for this specific aspect, we are currently collaborating with the team of Professor Kaznav Gassio, who works at Singapore Lipidomics Incubator. And his group has actually already developed the LICAR application, which works very nicely for the correction of the isotopic overlap using the MRM data. So this correction is specifically based on the transitions and not the precursors themselves, as we are used to with the high resolution data. And with this, I will finish. My special thanks for all of this work go first to all the members of my team here in Lausanne, and specifically Dr. Hector Gallart Ayala, who has really been dedicated for the development of this method, as well as the supervision of our technicians and students. So many thanks also to Tony Teeve and Rebecca Borgin, so our research technicians, and then our PhD students, Jessica and Anaï, who are currently working on the metabolic phenotyping of Lausanne population. Then many thanks go as well to our precious collaborators. So as I have mentioned, the team of Professor Kaznav Gassio, who is working on the application for the correction of the isotopic overlap, the team of Martin Gira at Leiden University Medical Center, who has contributed for the cross-validation with the lipidizer. Many thanks to the Thermo Fisher production team led by Claudia Martins, who for really very precious exchange during this method development. Thank you to Avanti as well for developing and releasing finally this this precious kit, and Christina Jones at the NIST Institute for providing us with the NIST plasma reference materials. I would also like to say many thanks to our medical collaborators, so epidemiologists and statisticians that are actually in the clinical consortiums of this two population studies, so the COLAUS, Complete Health and Heart. And many thanks to you for listening. So I just wanted to finish this presentation by providing you a glance of this beautiful view we can enjoy here in Lausanne on almost daily basis, if it's sunny, of course. And yes, to finish, I would just like to say that we still do enjoy untargeted, but we particularly appreciate the power of targeted and this in an absolute manner, of course. So yes, thanks again. Thank you very much for your attention.
All right, thank you very much, Juliana. That was a really interesting and exciting talk. We've had several questions coming in, so I'll just kind of go with those. It seems like we have a couple of questions about the choice of HELIC.
Yeah.
So I wonder if you could just elaborate on the choice of HELIC, specifically if the head group based separation is really needed for what you're doing.
Yeah, well, I would say that definitively with this method, this method allowed us to really expand significantly the coverage of the lipid we can profile. Yes, of course, there is an issue with the neutral lipids that are going out simply in the void volume in positive ionization mode. But as I specified, thanks to really this very, very high scanning speed of the TSQ Altis, I must say, we were able to obtain a very well-defined peaks that we have evaluated actually the precision of our method. We are even now evaluating it at a very large scale, so that we can really confirm its possibility for clinical applications. But yes, I must say that yes, thanks to Helic, we were able to actually significantly expand the coverage of the profiled lipidome.
Great, thanks. I also had a question about your extraction method. So was this the MTBE with methanol and water method, the Matiash method? Because we had a comment that there had been good experiences with extracting lysophospholipids, which I think you said you had difficulty with.
Yeah. Well, this is only, this is based on our own experience. Yes, I can say that it's not that we cannot get the signal for them at all. We can extract them, but the signal is indeed, I would say, much better when they are extracted indeed with isopropanol or BUMI compared to Matiash method. But it's not that we cannot obtain a signal for the lysophospholipids. I hope, you have understood what I meant. What I have shown on the spider plots was actually the relative abundance compared to the signal that was recovered with the Matiash method. So Matiash was always 1 as a reference on these plots. Okay. This was just to show actually that surprisingly, even to us, this single-step extraction was really as efficient as the biphasic extraction protocols. Mm-hmm. And this was quite surprising to us. And what I see as a real benefit is that it's very easy to actually automate a single-step extraction as compared to a biphasic extraction, mm-hmm. which is not, of course, impossible either, but it's just easier.
Yeah. So you are using automated liquid handlers for the extraction?
Yes.
Okay. Let's see. Another question was about acylcarnitines. If you considered adding those into the mix as well, because those are not currently in Splash. That's something that we do have in development, but they're not currently available.
That would be nice. We can definitively with this method nicely profile acylcarnitines as well, and we were thinking about adding them, to the positive mode method. It's just that we have already developed other. We have other methods set up for the measurement of quite a large panel of acylcarnitines. This is why it was not our priority, but they can easily be added.
It looks like another question just came in. We've moved to uniphasic extraction with IPA as well and found the same. There's no need for biphasic. So interesting. Yeah, so it looks like a lot of people are having that experience.
This was quite surprising, actually. Yes, it works really nicely. Otherwise I would not be promoting it so much.
Yeah. So no issues with, so the protein, I guess, is precipitated out quite well, and no matrix effects with that. Wow, it's very interesting. No. Yeah. I have just sort of a general question on commercially available sort of standard mixtures. And I was wondering if you could just sort of comment on how having these commercially available standards mixtures can sort of help democratize lipid screening and make sort of more routine quantitative workflows.
Mm-hmm. Yeah, I must say we were in the very beginning, we thought we will have to go with a lipidizer. because there was no alternative. And I must say I was very happy when Avanti released Ultimate Splash in the beginning actually of the last year only. Because yes, like you said, now we can actually in a way standardize our methods better. Everyone can use it. It's going to be much easier to cross compare our results, and I think this is now really essential. We have to be able to cross compare the results. This is also why I'm so very stubborn about targeted analysis and the acquisition of the quantitative data. I think it's really something necessary in metabolomics and lipidomics community. We have to be acquiring quantitative data if we really want to make sure that one day the metabolic phenotyping goes to clinics. So yeah, I think, yeah, this type of really kits are essential for further standardization and harmonization and cross comparability of the results. Yeah, it will facilitate this at least.
Great. Another, so maybe I missed this, but how absolute is your quantitation?
So our quantification is not absolute. I mean, if we cannot, I fully agree that this is based on a single point calibration. This is how far, this is, I think, the best we can do at this moment, because we are screening for an extremely wide panel of lipid species. So we have to, in a way, find strategies to be able to do this. So it's based on single point calibration, but as I have presented, we can, this type of calibration can be applied because the response is indeed nicely linear for most of the lipid species. As I said, we are currently evaluating the precision of our method, and this will be published for the entire set of lipid species. So I would say this is still, I mean, if we can call it an estimated concentration, because this is what it is in lipidomics. I mean, when we are targeting such a wide panel of species, if we focus on a specific, I don't know, 20 to 30 species from a specific class, then yes, we can absolutely perform the absolute concentration, absolute quantification using calibration curves for all of them. Yeah, such as for sphingolipids.
Yeah. And then sort of on that same point, have you considered using the Agilent robot to do automation of adding different amounts of splash to have a multi-point calibration?
Yes, this is quite, this is very interesting. Yes, thank you. Yeah. Not yet, but we might now. Yeah, no, we have been thinking about it. Yeah, but it's just the matter of time. Yeah.
Yes, yeah, of course. Yeah, I was curious. So you presented data on plasma, and you mentioned urine as well. Are there any other specimens or tissues that you're interested in going after in addition to those two?
Mm-hmm. Yes, we do function as a platform. So you must consider that we are analyzing a very, very wide panel of biological matrices. So we have already applied this method to different types of blood cells, to the whole blood, I mean the dry blood spots, to many different types of cells. So yeah, no issue at all. Different tissue types as well. So yeah, this is, I wanted to put the accent on plasma because of these population studies and because we are really, I would say, enthusiastic about clinical applications and would really like bring this metabolomics to another level with respect to actually the clinical applications. Yeah. Yeah,
absolutely. Does anyone else have any questions before we finish up? I'll give a couple more minutes just in case anyone wants to type in a question or unmute and ask themselves. But during that, thank you so much, Juliana. This was a really interesting presentation and really exciting data. And I think we're all excited to read the paper. Yeah, and we have another question which is rolled in. How about IPA for tissues?
Yeah, this is, we have tried. It works very well, very nicely on the brain tissue, for example, a liver tissue as well. Yeah, no issues to use it. I may refer maybe to some of the, I mean, I saw just the comment in the chat from Diana. Indeed, what we found interesting actually with biphasic extractions, that actually in the aqueous phase, we recover from biphasic extraction, we cannot detect so many polar lipids, polar metabolites, as we can when we do two subsequent extractions. So this is how we proceed for tissue. For example, when we are interested to extract from one same sample, both polar metabolites and lipids, then we usually apply like two subsequent extractions. So the first one with 80% of methanol, that is then we take an aliquot for the profiling, I mean the analysis of polar metabolites. The rest of it is pooled with the extraction, I mean the subsequent extraction with IPA for the lipid profiling. So this is just to say that we were also surprised to see this, that actually some metabolites that we consider polar, being polar or moderately polar, will actually partition, will actually be, let's say, will not necessarily remain in the aqueous phase only. This is what we could conclude from based on our experience. But yeah, maybe this merits more further experiments.
Yeah, very interesting. Okay, let's see. Before using the 1-point calibration to quantify lipids in samples, did the linearity of all internal standards evaluated by, I guess, was the linearity of all internal standards evaluated by multiple calibration points in matrix?
Yes, correct. It was done in matrix. Okay. This is what I don't know if you have seen on my slide, but we actually we used like a dilution series of ultimate splash that was evaporated, and it was reconstituted in plasma with isopropanol. And this way we actually obtained like a calibration curve, I mean calibration curves that were evaluated for these 75 different internal standards that, as I said previously, actually cover all different lipid classes, also with varied fatty acid chain composition.
Okay. If there are no more questions, then I believe we will go ahead and close it out, if that's okay with you, Juliana.
Yes,
So
thank you very much.
Yeah, thank you so much for giving a great talk. And thank you everyone for asking questions. These were very insightful. And thank you to MANA 2021 for letting us have this session. We were very happy to be here.
Thank you very much for listening.
Super, thank you.
Yeah. Hope everyone has a great rest of the conference.
Thank you, great talk, and nice to meet you. I've been seeing your name, so never had a chance.
Yes, me too. I saw it already previously.
Same.
You are also doing very nice work. Thank you.
Thank you. A very nice talk. Nice. Have a good afternoon.
Yes.
Bye.
Bye-bye.
All right, take care, everyone. Thank you, Juliana.
Bye.